Hello, in the last class I have introduced you different diagnostic techniques that are presently used across the globe to detect prostate cancer. 4 methods that I mentioned to you, and then we will come to our topic of discussion, that is imaging of prostate tissues using mass spectrometry platform. And when we are focusing on proteins to image, then we will use MALDI-MS spectrometry as an analytical platform. So, the main objectives of the present study that we are going to discuss about is to image prostate tissues from prostate cancer and also benign prostate hyperplasia. I told you that the prostate gland itself grows with age, is a natural process, and above age 60 the growth is meaning causes problem one after other problem related to urine issues and all that we described in the last class for men across the globe. Now, there are 2 type of growths. One is benign prostate hyperplasia, BPH. They are just it's just a growth, it's nothing to do with cancer. Another thing is prostate cancer. So, in both the cases, we will try to image the tissue using mass spectrometry and correlate our observation with the pathological finding. Pathological finding, remember, that is the gold standard across the globe. Mass spectrometry based imaging is emerging gradually. Obviously, in the future down the line sometime, I do not know exactly when it will happen, but it will come to the diagnostics definitely, because of its more specificity, less of its false either positive or negative results and so forth. But for the time being, we are just studying it for academic purpose, and if we get to see any scope opportunistically, then we will think of transferring the technology to the diagnostics. So, that is the main motto. Now, the objective 2 is molecular profiling of prostate tissue from prostate cancer and also BPH patients using mass spectrometry based proteomics. So, we will study both. One is imaging and another is proteomics, and here we are focused on tissue proteomics. So, obviously, patients meaning samples has to be collected from patients. So, ethical approval is required for the study to conduct. Then obviously, there must be inclusion and exclusion criteria, and no matter whatever patient samples we get, it has to be taken with prior consent from the patient. And obviously, before that the study has to be approved by ethical approval committee of the concerned organization. So, the inclusion criteria is patients present with prostate cancer and benign prostatic hyperplasia, BPH. The exclusion criteria is all cases of prostate cancer and benign prostate condition who have been diagnosed with any other cancer or with previous history of tumor surgery and or undergoing radiation or chemotherapy. So, this is the exclusion criteria. Sample size about 20 prostate cancer and 20 BPH tissues collected from patient samples, and these tissues were used for imaging, should be used for imaging and proteomics, tissue proteomics study as well. And the imaging that we will do using MALDI-MS spectrometry imaging, and though while talking about rat kidney tissues imaging in the previous class, I explained to you the protocol, experimental protocol for processing the tissues is very important, and it is not very common though, like regular mass spectrometry based proteomics or MALDI-MS spectrometry study. So, it is important to know the procedure, each and every step, why we are doing so, and what is the benefit of it, what the problem if we do not do this step, and so on and so forth. And obviously, I showed you the meaning rat kidney tissue imaging processing, and now I'll be talking a little bit about prostate, human prostate tissue processing for imaging. By and large, the procedure is same, but it's better to go through once as we are studying this. So, first and foremost is that collection of the tissues and then fresh frozen it. We will be talking about fresh frozen tissue section, not paraffin embedded formalin fixed tissues. So, FFP tissues we are not talking about. Because as I mentioned to you that the moment we put the tissue into formalin, that it leads to cross-link between different proteins present in the tissue, and moment that happens, then you will have problem to visualize proteins, because we will be visualizing protein by digesting in situ and then lifting different proteolytic peptides that are being generated. Now, if multiple proteins are connected by cross links, then it will not be easy to digest, as well as not easy to lift the proteolytic peptide from the tissue into the gas phase to study under MALDI-MS spectrometry platform. So, we will be talking about, or we will be working with fresh frozen tissue. Now, the thickness is 8 micron, roughly 8 micron thickness, micrometer thickness sections, and we will get the sectioning done in cryomicrotome, and then take thaw mounting the section onto glass slides. Next, this is next thing is to remove lipids and salts which actually interfere with the ionization of proteins or peptides from the sample. And this is very crucial step. If you remember, we did the same procedure in case of rat tissues also. So, the series of different solutions with different polarity that will take the tissue through tissue section, I mean to say, then 70% ethanol will dip it in for 30 seconds, then 100% ethanol, again 30 seconds, I forgot to mention here, and then Carnoy's solution, which is basically the composition I may explain to you. So, that has 3 solvents. So, within the Carnoy's solution we will put for 2 minutes, then again back to 100% ethanol for 30 seconds, then 0.25% TFA, trifluoroacetic acid in water, and again 30 seconds, and then 100% ethanol 30 seconds. So, in situ proteolytic digestion with the trypsin, once we are able to successfully remove the lipids and salts that are interfering agents with the peptides in mass spectrometry platform, then we talk about tissue digestion, and that is in situ. So, the main key point is we have to digest the proteins in the tissue itself without disturbing its biological location, heterogeneous distribution of the protein across the tissue section. That is very crucially important in tissue imaging step. So, for that we add trypsin and then 1% beta octyl glucopyranoside sprayed over the tissue section, a specific flow rate 6 microliter per minute, and 10 cycles using sprayer, automated sprayer, and then incubated overnight at 37 degrees centigrade for digestion to happen successfully. Next thing is matrix application. So, matrix we will use alpha cyano 4-hydroxycinnamonic acid, which is actually very common matrix. We have been using this in our discussion for all type of peptides to make it visible. So, then this matrix is sprayed over the tissue section at a flow rate of 10 microliter per minute for first 2 layers, and then 15 microliter per minute for next 6 layers, and then 20 microliter per minute for last 2 layers. So, 10 layers of matrix spraying we are talking about. Next thing comes is imaging to record. So, MALDI imaging data was recorded using HDI software, and then in positive ionization mode, range of interest is 1000 to 4500 m/z, because generally we do not prefer to record anything below 1000 m/z when we are using alpha cyano 4-hydroxycinnamic acid as a matrix. because in that zone this peaks from matrix will be filled up, that we have discussed many times. And then the scan time is 1 second in sensitivity mode. Here sensitivity is more important than to use very high resolution, and because when we are looking at peptides 1000 plus minus 1000 2000 Dalton, so if we have the mass accuracy after at the second decimal point good enough, and then we'll more interested on sensitivity, so you pick the very low intense also. And then pixel size, which is the additional thing in the imaging, is 90 micron for both x and y direction. Laser energy is 370 microjoule per second, and laser repetition rate was 1000 hertz. So this is the protocol that is used, and its detail explained in this paper. Now let us talk about imaging. Before I go to imaging, mass spec imaging, to show it is obvious to discuss about H&E stain image seen under the microscope, because that is the gold standard. So this is basically the prostate tissue that we are going to analyze, going to take on for mass spectrometry based imaging for different proteins to visualize. Now this is H&E stain tissues. So gold standard method, definitive for the definitive tissue based diagnosis of cancer is still microscopy stain tissue section read by a trained pathologist, and in this procedure of diagnosis, subsequent gradation of cancer based on the characterization of cellular morphology and alteration of tissue architecture in H&E stain tissue section is done, and that is the skill of a pathologist. It's a professional world, and then that's what like a mass, it's not a job of a mass spectroscopist or mass spectrometrist. So this is the reading and different labeling was done with the help of a pathologist. So this figure basically represents the digital image of H&E stain prostate cancer tissue section that was obtained after cryosection of cancer tissue core. So A is the total tissue, B, C, D, E, these are the 4 panels, B, C, D and E, these are 4 panels, represents 20x views of different region of tissue sections, showing distribution of tumor cell observed in variable amount of stroma. Figure B and C shows the lobular nest and acinar pattern of the cancer cell separated by fibrous stroma inflated with the scattered lymphocytes. So this labeling obviously is completely pathological term, and mass spectrometrist has nothing to do with it. So I am stating this as it is labeled by the trained pathologist. So these are the different, but still if we look at, we can see like this, the cancer cells are all these are cancer cells. By the way, here cancer cells are focused at, nucleus is larger in size, and these are the cancerous prostate cells, CPC. Here these are the normal cell like is encircled here, and then this is extracellular matrix. Now if you look at this zone, which is here, it's enlarged portion, it is filled with cancer cells, right? Now so these B and C, these portions, if you, that is the blown up picture, it's very clear that it's filled with cancer cells, and then definitive pattern by which cancer cells are arranged in the tissues, like, and accordingly the names are there, like nest. So here the cancer cells are appearing as nest here, okay. So as the different names that are there, acinar, lobular and nest pattern of the cancer cell arrangement. Here in C, lobular pattern of cancer cell arrangement separated by fibrous stroma inflated with scattered lymphocyte, okay. Now in D and E shows arrangement of cancer cell in acini and glands, like D here. So these are basically cancer cells, and E, these are again cancer cells. So this picture is taken from this paper. So you can look through this if you are interested to learn in detail. So next job is the mass spectrometry that we are planning to, meaning we are our main theme to discuss it here. So this spectra, basically the combined MALDI-MS spectra obtained from all pixel points within prostate cancer tissue. So entire tissue section, the one that I showed, the serial section of it is actually scanned, and this entire tissue section gave the mass spectra, obviously with the information that is kept on background, 90 pixel in X and Y directions, and then this spectra, the one that is shown here, is the summation of all firing that is done at different portions of the tissue. So this is why it's called combined mass spectra. Generally in tissue, the background noise is heavy, which is very clear here. Okay, now next question to see that how many signals are there. So if you look through the list, obviously we didn't able to label all the things, but if we look through the list, then the total is 90 proteolytic peptides. These are all peptides. Remember, we actually went for in situ digestion, and then the tissue was fired with laser. So whatever we are getting it here, all peptides generated from different proteins sitting across the tissue. So this, if we sum them up, then its total is 90 proteolytic peptides obtained from the observed in the mass spectra. Okay. Now the next step is very crucial. We have to do proteomics, because we need to know identity of these peptides. Literally, we can do it from the tissue itself if our MALDI is very sensitive one. And honestly speaking, these low intense peaks is very difficult to get the tandem mass spectra done. So until unless we have the good tandem mass spectra, as you know, we have discussed a lot about de novo sequencing in the very beginning of this course. It's not easy to sequence them. So we will not be able to identify and characterize them. So for that we need to do proteomics. So and for that we have to start with a separate tissue section, serial sections maybe, or a lump of tissues that we will class and then extract the protein out. So the steps are: the lyophilized prostate tissues were classed to powder, and it was suspended in lysis buffer, because we have to lyse the tissue. Protease inhibitor cocktail was added and subsequently sonicated. Okay. Now suspensions was centrifuged and supernatant was dialyzed overnight against 50 millimolar ammonium bicarbonate, pH 7.4, followed by concentration measure, and then in solution digestion. So moment we do in solution digestion, then only it's possible to go for proteomics, meaning we are talking about bottom up proteomics. So sample was denatured, because we have to denature the protein samples, and that is done by a mass spec compatible denaturant, 0.2% rapidest at 80 degrees centigrade for 15 minutes. Disulfide linkages should be reduced and alkylated. Then trypsin must be added for digestion. So trypsin is added to the protein extract with an enzyme substrate ratio 1:10, that is the standard one. Incubated overnight for 37 degrees centigrade. Sample was acidified with 0.5% formic acid and incubated at 37 degrees centigrade for 90 minutes, and that to facilitate the hydrolysis of rapidest detergent, because we have to destroy it before we take it to mass spec. That we have talked about the same thing previously also. Samples was centrifuged, supernatant was collected, and then we have to spike with a standard, that is enolase digest, and then sample was subjected to nano-LC SIMS. So what do we do out of this? Upon analysis of the tissue proteomics data, 52 proteins could be identified. Though we have seen 90 proteolytic peptides, and obviously 90 from, we hope that all of them has been characterized, and then tissue proteomics data showed total of 52 proteins. Okay. Now, intensities of obviously of those 52 proteins are plotted along XY coordinate, retrieving the information from mass spectrometer that was meaning that is kept while recording, and subsequently image of the distribution of these peptides were obtained in the experimental tissue section, which is what actually MALDI image. So, in the MALDI image 41 proteins were observed with low sensitivity that their differential spatial distribution was unclear. So, then discarded. So, 41 gone out of 52. Now, we are left with 11. Now, out of 11, 8 proteins were observed to be having uniform pattern of distribution across tissue section. That means, though the tissue is cancerous, but they are uniformly present. So, these were characterized as fragment of trypsin, which was used for proteolytic digestion in situ, hemoglobin, and also isoforms of cytoskeletal protein, actin and myosin. So, those will be there across the tissue section, no matter the cell is cancerous or not. So, those 8 gone. So, 41 plus 8 gone, 49, we got we got 52. So, we are left with 3 proteins, and the remaining 3 proteins were of protein of interest, as their differential distribution across tissue section could be attributed to prostate cancer. So, we will study these 3 proteins in detail, but before I go there, let us talk of at least one example which is uniformly distributed. So, this figure shows homogeneous distribution of peptide with M by Z 1455.8 across the experimental prostate cancer tissue section in mass spectrometry based MALDI imaging. Nano LC SIMS based tissue proteomics identified this peptide as a tryptic fragment of a cytoskeletal protein myosin 11 spanning residue this. Okay. So, this is the MALDI tissue image, and this is histopath image of the serial section. So, figure A, this one shows the corresponding representative regions in the H&E stained tissue section, that is different encircled regions, okay, where myosin 11 was obviously was observed as homogeneously expressed in the MALDI image of the serial section of the same tissue as depicted in the figure B. Okay. Now, encircled portion like the one that we have here and here, this encircled portion basically represents different region of tissue section showing similar signal intensities of the peptide 1455.8. So, they are basically similar, and in here more intense, in the entire zone the intensity is almost same, in the entire zone intensity is almost same, and so on and so forth. Okay. Figure C, this is showing the isotopic distribution of the peak 1455.8. So, this is monoisotopic, then 1 C13, 2 C13, 3 C13 like that. We know that, we have studied it in detail. Now, figure D and E, these two are actually representing 20x expanded view of small representative part of the encircled areas, showing population in this region, how the cells are populated, the cancers and all. So, upon analyzing the cellular population in the H&E stained tissue section, we observed myosin 11 was homogeneously distributed in this region, having high density of cell as well as other region having greater amount of stroma with immune cells. So, it is basically distributed all over the places, but in this region it is high, in this region all are same, almost same in intensity. So, what is its function? It has been reported that myosin 11 is expressed in smooth muscle cell of the prostate gland. Now, its major function is in the smooth muscle contraction upon association with acting microfilament proteins. Okay. Now, additionally, in prostate cancer tissue, smooth muscle cells are also contributed by the new blood vessels that is formed during angiogenesis. That means the process of formation of new blood cells through the tumor, so that the blood, meaning those cells gets nutrient, and whose fine branches can spread across the entire tissue section. So, therefore, uniform distribution of myosin 11 across the tissue section might owe to the above factors, which is what we are explaining here. So, as the tissues are, meaning cells are growing, so then obviously they will have the cytoskeleton protein myosin 11 visible or expected to be visible all over the places. So, therefore, this observation correlates well with the reported cytological features of human prostate cancer tissue. So, this is what the tissue image looks like, which you have seen previously for rat tissues. This is human prostate tissues, and one specific peptide, obviously that represents one protein, how it is located across this, and this is basically heat map, where black is basically background, then blue, then red, and most intense one is yellow. So, this yellow distribution of yellow represents here that it is more concentrated here, which we explain here also. Now, next thing I will show you how the images correspond to those 3 proteins that we said that out of 52 proteins, 3 we found to be interesting, and that is what we will describe that, but out of, meaning for the sake of time, for the interest of time, I have to close the class here itself. So, in the next class, I will describe the image of those 3 proteins that are very much important related to prostate cancer. Thank you.